We're going to start off this afternoon, this session, with a couple of panels on some, I think, some pretty important and some pretty relevant topics for a lot of people in the room. So for this first session, I've got a dual role, so luckily I don't have to introduce myself again, but we do have joining us remotely Iona Macleod, who will be presenting, and we'll also have four tremendous panellists that are going to be with us. So first of all, what I'd like to do is just introduce Iona, who is a senior research geneticist with Ag Victoria, working in the dairy bio team. She grew up on a small mixed sheep dairy beef farm in north of Scotland, which you'll never know that that's the case, and one day and hoped to take over. Instead, she ended up working in agriculture in Thailand, Cambodia, Pakistan, as well as in West Australia and Victoria. Since 2003, she's been working in the dairy cattle genomics with the Agricultural Victoria team, and where also she undertook her PhD. Much of her current research focuses on how to exploit genomic data to bring accurate genomic breeder values to all breeds and crossbreds. So really doing some important work. So before Iona does her presentation, I'm going to get our panellists up here so that way they're ready to go when we need to start the panel. So it's a bit of a privilege to call up these four people in many and very different ways. So I'm going to start off with Paul. Paul is involved and committed into the Aussie Reds, was involved very early, has been a wide cross-section through doing some work in stock feed companies, also with herd improvement, works on a 700 cow herd, which within that herd has his Paco Aussie Red herd, which has been ranked either first or second in Australia on BPI. And the other part, I think, that's really important, Paul's somebody who, within the Aussie Reds, not only enjoys the breeding aspect, but contributes to the association. I think it's treasurer at one stage. And
no, just the technical.
Technical. So the good part is that, you know, it contributes. The second person I'd call up is Trevor Parrish. Trevor, with family, has been a stalwart of the Kangaroo Valley. And if anyone has the opportunity to go to that beautiful part of the world, maybe not drop in on Trevor, just depending on the time of the day and whether you catch him. Sometimes you refer to people as, godfathers of this. I think Trevor, in some respects, like a godson of genomics. I think he's up to about 6 generations in the herd that have been tested, and has been somebody who has always taken on I think technology not only to improve breeding program, but also to profitability and decision making. So Leah's here. I'm not sure if she's in the room.
She's been shopping.
Oh, shopping. Okay. There's no wonder you need more profitability. So it's a family operation and continuing to expand. So welcome, Trevor, to the group. The next guest is Sandra Bacon, Brookbora Jerseys, with husband Robert, son Daniel, and a new addition Stacey. They run an operation at Tennyson in northern Victoria, focused on, well, I'm biased around this. I think one of the best Jersey herds in Australia, and stand up as one of the best herds in the world. I really think I had the privilege of being there. So one of the things I find interesting around this, because Daniel's also works as a nutritionist, so not only do they run great cows, but they're great farmers, is that as we go on, how to take a wonderful group of cows where they've also, not only with production, but they've been Premier Breeder, Premier Exhibitor at International Dairy Week, and really have set a very high standard for the quality of their herd. And the last, and certainly not least, and this fits in exactly the same picture as Lyndon Cleggett, who has set a standard within the Guernsey breed for many, many years, having got involved in Guernseys. I think he inherited a little bit from his father's passion around Guernsey cattle, from Langhorne Creek down to Mount Gambier. milk around, converted the beef and sheep property, milking about 300, has been such a strong supporter both in Australia and internationally in Guernseys. Also has got a next generation that's contributing to that. Lyndon and Joyce, his wife, have been successful right across the board in respect to showing and just breeding outstanding cows. And the other aspect is that Lyndon also served for 7 years as a director for Australian Dairy Herd Improvement Scheme, ADHS, the pre-runner to data gene. So it's not just what happened directly in farm and the Guernsey breed, but also the wider industry. So I'm really honoured to have 4 guests that we have on the panel today. So I'm now going to hand over to Iona to present her work. Thank you.
Thank you very much, Graeme, and good afternoon to everybody. And thank you again to the organisers for the opportunity to just give a quick snapshot of some of the work that we've been doing at Dairy Bio. Looking at the title of my talk, there are a couple of things I want to stress before I get started. And the first is that when I talk about smaller breeds, what I really mean is not small in stature, but ones where the numbers of animals in Australian herds are much less, fewer than the Holsteins. And the second thing that I want to highlight or emphasize is that while I am tending to focus through the talk on bringing genomics to small breeds, I am emphatically talking about bringing improved genomics to all breeds. So the things we have done and things that we'll focus on today are helping Holsteins as well as many of the other, as well as other breeds of dairy animals. So I just need to make these clickers work. So that seems to have worked. So just quickly summarising some of the key benefits of genomics to set the scene about why we're doing some of the things we're doing. A very obvious one is parentage confirmation, and that's really now available to all the breeds already. And it also relates to our ability to use genomics to predict breed proportions, which is also of interest for a range of reasons. Today, I want to focus mainly on that second and third topic there. The second one is often spoken about as major genes, and these are just really mutations, as we call them, that have a large effect, and they could be good or bad. So things like the jumps, poll versus horn, and so on are major genes. So I'll spend a bit of time talking about them, but also mainly about the third topic, which is providing genomic breeding values in young animals that are reliable enough to use for genomic selection or for increasing genetic gain, or, for example, to use in cows for management decisions. So many of you will have seen probably this dairy biographic that shows the improvement in reliability of Holsteins for the milk traits over time. So starting out in 2009, going across 10 years to 2019. And clearly, obviously, nearly doubling in that period of time. But if we look at the graphic for fertility, while there's been an equally impressive doubling of the reliability across that 10-year period, we can see that the reliability is much lower for fertility. And so the question is, what's driving this difference between milk traits and fertility? And the first and most important driver for this difference in reliability is the number of genotyped animals with accurate records. So for the milk production traits, these are much easier to record. We have many more accurate records. And vice versa, in fertility, it's much more difficult to get good records. There is another factor that also has an impact, and that is the genetic variability or the heritability of the traits. So fertility has a much lower heritability compared to milk production. But that's not something that's within our control. So we want to focus on things that we can control today, and that is the size of our reference populations. So you've heard a lot already today about how we need large reference populations for genomics to work really well, to get high reliability. That means genotyped and recorded animals. So what do we do for small breeds? That means we've got a major hurdle to overcome, because small breeds, by definition, we can't get a large reference population. So some of the things that we've been paying a lot of attention to are how to develop approaches that use information across breeds, and also, of course, developing collaborations across countries. We have spent also quite a considerable amount of effort on looking at improved genomic selection methods. And then the third topic here is the one that I'll probably focus most on today, where we've spent a lot of time in the Dairy Bio Project 1 trying to develop SNP chips, or a set of markers that give us more robust and reliable genomic evaluations. So to find good genetic markers to put on SNP chips or to use for the evaluations, we need things that are cheap and robust to genotype. They need to be able to be accurately imputed up to the, to the standard set of markers that we decide are going to be used for the evaluations. And they've got to be the best markers available for genomic evaluation across all the dairy traits, so not just for one or two, but for all traits. So we've put a tremendous amount of effort into doing this in the Dairy Bio Project. So sorry, I just yep. So this is where the 1,000 Bull Genomes Project comes in, of finding better markers, so better markers for our genomic evaluations to make them more reliable. And what we do here is, for whole genome sequencing, essentially we're getting the information on every position across the whole genome of cattle. And we started out thinking it was very ambitious to aim for 1,000 sequences across bulls' genomes, but in the end we're actually now up at about 5,000 animals. So this is an expensive business. While sequencing has become a lot cheaper over the years, it's still more expensive than doing a genotype for a SNP chip. So this is a huge collaboration across countries. We've got about 40 partner institutions, 20 different countries, and over 200 breeds and crosses of cattle are involved. On those 5,000 animals, we've got about 3 billion letter code for every animal. So that's big data. So we've been talking about how we use big data in our lives at the moment. And essentially, the most important thing about this 1,000 Bull Genomes Project is that what this allows us to do is it allows us at Dairy Bio to impute a virtual sequence into every animal that has a SNP chip genotype. So that means many of you out in the audience there and people on our panel, we have animals of yours which you've genotyped, and now we've imputed whole genome sequence into those animals. So at the current count, we've got over 200,000 Australian animals, and we've done many more for our collaborators too, but over 200,000 Australian cattle have virtual sequences that we're using in our research. Once we get that virtual sequence, then that allows us to do hundreds of thousands of different analyses with that data across all the dairy traits that are important currently to the dairy industry, to work out which of those many mutations, and there are about 30 million mutations across the dairy cattle that we're looking at, that differ between animals, that may have some impact. And our job is to try and work out which of those 30 million variants or mutations are interesting for the traits that we work with. So in addition to all the data that we get from the 1,000 Bone Genomes Project and the imputed data we get on those 200,000 animals, we're also involved in doing other special sequencing methods where we're really trying to home in on what are the functional mutations in amongst those 30 million letters or mutations that differ between animals, which of those actually affect the traits that we're interested in. So while there are many hundreds of thousands of mutations that might affect each trait, we think, and some of those are overlapping between traits for things such as fertility, milk yield, and so on, that means that there are still millions of those mutations which aren't actually going to be useful for us to use for the evaluations in dairy cattle. So we're trying to sift out between the useful ones and the non-useful ones. And so another thing that we undertake is looking at sequencing methods that allow us to look at gene expression in lots of different tissues across the animal. And again, you know, this is difficult and very laborious work when you're looking at lots of different tissues, gene expression. But the critical thing here is by doing that, it allows us to find the actual, so we know a lot about genes and mutations inside genes, but what we know much less about, and this is true of the human field too, and they're mirroring the exact same work that we're doing, what we don't know about is what makes a gene get turned on or be more expressed, so switches on and off, and also dimmer switches that say express this more or less. And these are very important, we know, to all the traits that we're interested in the dairy industry. So we undertake this work as part of a global collaboration called the Fan Consortium. So that consortium stands for the functional annotation of animal genomes, but it essentially has allowed us to get much closer to what the real mutations are that we need to use for the SNP chips that we're designing. So that's just a little cartoon image of what it is we're doing. So we're looking at gene expression across many different tissues, many more than I've pictured here, and looking for these switches that are switching on or off the expression or dimming down the expression of genes. So where are we actually at? Well, currently in the Australian dairy industry, what is used for genomic evaluation is what I'm calling this standard 50K chip. And without going into the details of the cartoon, we have now developed, using the information from the Dairy Bio Project, what we call the custom or the AgVic custom XT50K chip. It's a much higher precision SNP chip because it's much closer to causal variants. And if we focus in on that table on your right-hand side, this is comparing the increase in reliability that we get in genomic evaluations. This is averaged across milk traits. When we use this new custom SNP chip compared to just using the current industry 50K SNP chip. And what you can see is that even for Holsteins, this gives a lift of 2%, 3% in Jerseys, 9% in Australian Reds, and another 7% lift in the New Zealand or Kiwi crosses. So essentially what this shows, and it's consistent across all the traits that we've tested, is that for the smaller breeds, we get the most benefit from this kind of an improved SNP chip. But even for Holsteins, it has a benefit. So where are we at in terms of what's happening in the industry? Well, I think it was alluded to, I think Jackie mentioned it today in her talk, that all this research-based evidence from the Dairy Bio Project, we've worked together with DataGene, and now the all the background research that we've done is being used and being put into practice to develop the Australian red genomic evaluations. And it's really pleasing to see that they're going to be utilising, for the first time ever, this new custom XT50K SNP chip that we've designed and developed. And they're also using an improved genomic prediction method. Furthermore, we've come up with improved methods that are not only good for smaller breeds, but also crossbreds. I just want to also mention that. So these, you may have heard Iona talk earlier about her work in the Australian Red, where she's demonstrated that by getting an optimum proportion of mixed breeds in her reference set, she can improve the reliability for the Australian Reds. But if you let the reference population be over-dominated by Holsteins, it actually then detracts from the gain that you would otherwise be getting. So we've done a lot of work, not only for Australian Reds, but also in crossbreds to demonstrate this, and shown that we can get an up to 9% increase in reliability by getting a more balanced mixed breed reference set. This is also in crossbreds is a way that you can actually exploit the crossbred data. And it can be exploited not just for crossbreds, but also for prediction of the smaller breeds. So if you've got crossbred jerseys, that will also help increase the reliability of purebred jerseys. We've been lucky enough to do the crossbred work because we've had a collaboration with New Zealand, where we've had access to over 16,000 New Zealand crosses. So one of the things I should emphasize that in our research on helping smaller breeds, one of our problems is that in order to do this, we need access to data from the smaller breeds to demonstrate that it works. And so it's a catch-22 that until we have the data, we can't demonstrate that it works. But for smaller breeds, they say, what's the point in genotyping if I'm not going to get genomic evaluations? So that's a tricky one. Sorry, I'm seeing I'm a bit behind in my clicking of the slides. I apologize for that. So I'll try and keep up. So this is the second last slide. And I just want to here just quickly draw your attention to the fact that we're not just talking about genomic evaluations, but also the fact that the 1,000 Bull Genomes Project is incredibly helpful in accelerating the rate at which we can discover genetic defects. And there are many examples, but I've just listed a few here, like the Holstein haplotype 1 that originated, we think, in the famous bull chief, and was responsible for an estimated 500,000 abortions before it was finally nailed to what the causal mutation was. The 1,000 Bull Project allows us a means to validate and trace these genetic defects, and I hope in the future that it will allow us to potentially impute these genetic defects, the mutations, into genotyped animals. So animals that are already on the ground, genotyped with, say, a standard SNP chip, where you don't want to go and pay for yet another genetic test because suddenly three new genetic defects have come out in the past year. Then if we can impute that into the data for you, that then avoids the need to go for more genetic tests. And how can we help identify new genetic defects? Because these are arising not rapidly, but they do continually run the risk of arising in the population and spreading very rapidly as a result of AI. So the way everybody can help is by recording and reporting things, and potentially, if possible, taking DNA samples. So just finally on the last slide, and I apologize. So looking forward, what's going to happen in the future in terms of all this genomics, it's very difficult to say. But some of the things that are up and coming are things like skim sequencing might be going to be replacing SNP chip genotyping. we're very excited in the research side about long-read sequencing, because that will allow us to detect some complex mutations that at the moment we can't detect with the current type of sequencing that's done in the Thousandable Genomes project. And we think there's also going to be new methods coming on board to better identify these functional mutations. And finally, there's obviously, going to be new and improved ways to use genomic tools. There might be new traits coming on board that we'll have to keep on top of. And these might include things like even some of the work we've been looking at on oligosaccharides, which have huge benefits for neonates, for babies. They're actually added to formula milk that's made from cow milk, because in human milk there's a much higher level of oligosaccharides. And we've demonstrated that these are very highly inherited genetically. And so there are things like that that may come to the fore in the future. My final slide is just to reiterate some of the key messages that we've developed a custom SNP chip. It's given us up to a 9% increase in reliability for the Australian reds. The second thing is that we need quality data, and by using a mix of breeds, we've been able to get up to 9% increase in reliability. That's for the crossbred cows. And that strategic collaborations have enabled us to improve our genomic tools. Finally, I want to acknowledge all the organizations and people who've been involved in funding, gathering data, and so on, particularly to GInfo and the farmers, all the farmers out there who are recording and providing information to us through DataGene. And I want to also apologise. It's always a danger when you put up acknowledgements to forget somebody. The group I have forgotten to include on this slide are the molecular genetics, animal genetics team at Dairy Bio, who perform a huge amount of the work for us on the sequencing side. So thank you to everybody. I think now Graeme's going to move into the panel discussion. I'm a bit worried because although I can't exactly see what's going on out there, it looks like you're probably all going to get a crick in your neck if you have to look up towards the screen. So Graeme, maybe I take the privilege of asking the first question, if that's okay.
That would be great, thank you.
So maybe I just kick off then with Trevor, if that's all right. And Trevor, my understanding is you were a very early adopter of genomics. So could you perhaps tell us about some of the significant benefits that genomics has brought to your business, and also maybe touch on some of the future advances that you see coming along your way?
All right. Originally, we probably tested with Genetics Australia some young bulls, and it moved on from there that they'd pay for doing a number of heifers as well. And that was I think in 2012, around that year, we thought, well, it was a great idea, so we'd do the whole lot of the heifers that were born that year. And since then, we've been doing every year, doing all the heifers. And it was surprising the number of mistakes that we'd made. When you thought you had them, 2 cows had calved, one was a black cow, one was a white cow. And you think that's what the sort of calves they'd have, and it'd come back and it was wrong. And I was sort of thinking, oh, all right, how many mistakes can I make? But I do make quite a few. And we picked up, once we milked some of them heifers, I noticed one particular heifer was very slow, which I didn't actually look on the genomic test. Looked at the genomic test, and she said she was well below average for milking speed. So it had already told me that it was actually going to be slow, but I didn't realise until I looked at the thing that, yes, it was right. I thought, well, this is pretty good. Lucky enough, those genetics were fairly well tested in Australia and by proven bulls. So as you go along, you use mainly proven bulls. Well, I was more excited to use some of the young genomic tested bulls and thought, well, let's try some of these bulls and see if they actually get the same results. And over time, we've probably did more and more of genomic bulls for size selection. As far as health traits coming along, like daughter fertility was always a bit of a worry. And I reckon now we don't see it as bad as what it was. We've bred for daughter fertility for a number of years. I'm probably looking at daughter fertility bulls now are 110, because I don't think 100's really an improvement. Probably 105 plus before you see a real improvement in Holsteins. We've done very well with the number of bulls we've been able to test at GA. The fact that you get to look at that information very early, and the fact that we actually test them from, say, could be a week to a month old. You know which bulls to now to keep them as a breeding bulls for other people to sell, or we just turn them into steers. So you sort of, they might be all right for a number of things, but they might be very bad for temperament, which we don't put up with. And there's little things that you look at and go, people want good bulls. We also at the time, they were starting to get the A2 testing. So we get them, everything's done for A2, BPI, the Australian system, the American system and the Canadian system, which comes back through Zoetis and Search Point, and we can actually select anything from the Search Point's point of view. It's a really good system. It comes up with polled, A2, a few of the health traits that we don't get, you can get from the American system or the Canadian system. They have LDAs, ketosis, so it's given me a variety of things to select from, and I'm thinking the Australian system as we go forward, them health traits will come into our system. So at the moment we hadn't tested for a little while, and we sent off 70-something other samples the other day, and we're sort of looking forward to them coming back. But now the move to weekly testing means that you sort of get them and you go, oh, all right, that's not so big change. But as they add more information and reliability to the system, it's just sort of, yeah, I just love looking at it occasionally. Sort of don't try and look at, we just basically just look at the lower ones and either made them to beef, which might sound funny, but it's being more of a trend. So, yeah.
Thanks, Trevor. Lyndon, perhaps at the current situation at the other end of the scale, the Guernsey breed in Australia doesn't have genomics available to it. So could you perhaps take this through how the journey may start and what the thoughts of yourself and the Guernsey breeders in Australia and internationally?
Well, we're desperately been trying to get the genomics up. And as a small breed, you've got to work internationally. I'm hoping we're not far away joining the US, as they do have genomics going in the US. Australia depends a lot on the US for genetics, so we have that ability to use their genomic testing now. And probably it runs on, in this breed in the last 15 years, the sire selection is a chill and kill, so proven old sires hasn't been a thing that we've had much access to in the latter years. And I think that's been pretty good anyway, as we keep moving on to the next generation. But I'm excited by hoping to get the genomics going here, because like home in my herd, I will be testing the young stock, breeding the young bulls. I mean, we AI made our heifers twice, and then we put a young sire out with them, and I'll be able to make sure that I'm putting the best possible sires with them. And I'm really want to get down that path as fast as we can go, and hopefully it won't be too long in the future. But also, if we can get genomics up in this country testing, it'll give the breed an opportunity to sell more genetics overseas as well, as there are cow families being seeded. But we need some measurements to take back for overseas countries to see what we've got in this country.
Okay, thank you. Do I turn over to you now, Iona, if you wanted to?
Okay, thank you. Sandra, maybe if I could ask you, you already have a very successful breeding program, so maybe you could tell us a bit about how you've utilised genomics to add value to your breeding program, and whether there have been financial benefits for you.
Yeah, I guess for us it's a piece to a puzzle. We use it with our production, with classification history, with pedigree history, and an independent fresh set of eyes just to look at our herd. But we use it, we've got all the genomics of the herd along with all the information on a spreadsheet at the dairy, so when we're joining it, they are AI bench, pardon me. The computer's there, we can scroll through it. All the strengths and weaknesses are highlighted so that we can actually look at what we're doing with every cow. I guess we're not milking 1,000 cows, we're milking less than 300, so we've got a bit more hands-on with what we're doing. Using genomics over the time, it has shown the trends, how we've been directing. For an example is, say, temperament has improved because we've been fairly hard on that. We don't like bad-mannered cows. Probably cell count, that's over time has come quite low, and now our average is well below 100, so it's worked over time. Over time using genomics, because we've got the whole herd done now, it's highlighted cow families that we've known. It's also highlighted a few of the new families, how they're reacting. With the financial benefits, that's more difficult to put a finger on at the moment, I suppose. With females, if we're selling individuals, it's more a whole part of a whole package rather than, because when we sell an individual, it's a package, not just, high this or high that. With bulls, it's been good. Again, with like the females, it's highlighted families, but it's also highlighted some new ones where some potential bull families have come to the top, I suppose you'd say. And at the end of the day, having classification across the generations and multi-generations, I guess is hopefully adding value to the herd, which looks better on the balance sheet, which hopefully the bank likes. So, because at the end of the day, that we're all in business. So that's probably where we're using it across the board.
Great, thanks. So Paul, Aussie Reds, red breeds, and I'd say because you source them from many different countries and combinations. So for you, the breed's been a big driver to get genomic breeding values. How's that journey been, and where do you see these steps going? As Iona has already put to us that progress is being made.
Yeah, no, it's been really exciting. The last 12 months, I suppose. We're sort of going back six years ago, I think it was, where we saw what was happening with Holstein and Jerseys, and we just thought, as a breed, if we don't do something, we're going to be left behind. We've been really on the front foot. Seeing at the same time the genomics in the red breeds in Europe being successful, and we've, as a breed, we've been utilising those genomic bulls from Europe, but you're still getting the varied results. So some are going to be successful, some have been disappointing. So, just having some more clues on how those bulls would perform in our environment was something that we really wanted to tap into, and that then flows into our own breeding programs. In the past, the Aussie Red Young Sire program that GA ran was, really going gangbusters, but it was, the bulls were selected the old-fashioned way, and, I'm sure some of the best bulls were being left behind in the paddock. Without that genomic information, it was, you're just shooting blind. So hopefully moving forward, we can select the right ones, or have a better chance of selecting the right ones. And for us as a breed right now, that is probably very critical. We're seeing more interest in our genetics from Asia and South America. New Zealand are even, interested in what the Aussie Red pasture-based sort of genetics can do in their system. So we need to continue to supply that quality product, otherwise those markets can peter out.
Okay, so pretty important.
It's very important, yep.
So, Iona, have you got any other questions or any other questions you'd like to raise to the group?
Yeah, so I just following on with Paul on the Australian Reds, I mean, it's, it's obviously a work in progress. We're not fully there yet. Are there any sort of particular challenges that you see that we need to still get on top of in terms of your Australian Red genomics?
Yeah, I think because we're such a small breed by population, we're never going to have the reliability of what Holstein's going to have. And with our genetic diversity within our breed, and I think the Australian red breed would probably have as much genetic diversity as even the Nordic breeds. You know, there's Illawarra background in the pedigrees, there's Ayrshire, there's New Zealand influence, and we want to protect that genetic diversity because I believe the genetic diversity is the key behind the benefits of our breed with regards to health and fertility and those sorts of things. So as we rely more on genomics, that increases the risk of us increasing inbreeding, which, you know, could have detrimental effects that we're not going to see until those things are expressed in the field. So yeah, I'm sort of hopeful that as a breed it won't develop into like a Formula One race where all companies are trying to get the #1 genomic bull and the breeders want the #1 genomic heifer. It'd be take a more balanced approach and just make better decisions, you know, on farm and with bull breeding.
Yeah, and maybe I should just add there too that, I mean, all that work that we've been doing, some of that I highlighted today, looking at also working with where we've worked with a lot of crossbred data, that has huge implications in terms of helping us work out ways to incorporate Illawarra and you know, Ayrshires and others within the red breed evaluation. So that's definitely something that going forward, I would very much hope that we'll be able to get better at and improve the reliability for all of those red breeds together.
Yeah, and I guess the other thing too is, you know, there's a lot of businesses out there that are utilising the three-way cross system that and doing it quite successfully. The ones, yeah, there's a few that do it well, and there's a missing link at the moment in that chain for those guys because the reds are a critical rotation in that strategy. So at the moment they can't tap into that power of genomics with, you know, you just imagine being able to select heifers with the production of a Holstein and the components of a Jersey and the health traits and fertility of a red. It's sort of pretty exciting for those guys too.
Yeah.
So I've got a question, and I'm going to paraphrase it, Trevor, because I want to direct it to a couple of members of the panel. Sandra, you basically touched on it about finding new cow families, and Trevor's the same. So through the process of testing over a number of generations, has there been surprises around some of the, because you do have such a focus on cow families, that it's changed some of your emphasis on what were some of your best cow families and some that maybe not as high up the food chain?
I would say it's confirmed some of the cow families that we've got. It's confirmed some of the cow families, our original ones that we've thought were just quietly just cruising along the bottom. It's probably confirmed that. They're just doing enough to tick along rather than... probably the other thing is we've brought families in from various herds over time, and it's particularly some of them, it's confirmed that they are very good families regardless of the management of where they are. It's been an interesting... Yeah, it's brought up a few families that we've purchased that have just been ticking along, and now particularly one family, it's sort of come to gradually just stepped up, and I guess we knew, and genomics has shown that what its potential is there, but it comes down to what bulls you're using and getting it right, which is not always that easy.
So Trevor, have you had any in those generations, anything that's jumped out at you that you go...
Yeah, well, it's funny because if we look how many, well, we milk 200 to 250, 260, but we're probably at 200 at the moment. We sell off just sell off the bottom end, but we've had one family that goes back to Laurie Wells, which is quite ironic that my father, when he originally sold Laurie Wells, I think he used a bull from Laurie and a cow family, and the bull was maybe last for Laurie, but the family goes back to this cow family that he got from Laurie out of a bull that was maybe last, and it's thrown up a lot of good bulls, but not quite on the type side, but the production and the health rates have all just stayed there. And there's another one where my father bought a cow family, and I've worked with the family for 13 generations of a classifier many years ago. I think his name was Eric Armour, which is probably most people wouldn't even remember him. So it's part of the sort of a thing that you remember these cow families are coming through with, and I think the 4 bulls we've got went to GA this year, they're out of 4 different families. We don't do any embryo transfer. We haven't done that for, I'm thinking nearly 10 years. So we did tried some IVF with some heifers to GA, but didn't get any results for that. But we just, I don't like needling cows that much. We're trying to use less drugs, so we're trying to go down that less drug-free path. Yeah.
Okay. Lyndon, there's one thing you mentioned, I just maybe explain a little bit, was the chill and kill aspect and the use of young sires, which is probably more unique from what you're doing, and perhaps there's a starting thing for using young genomic bulls as more information. Just touch on that just for a second, what you've done over the last few years.
Basically, a young bull only lasts in our herd to be used for 12 months, and as they just not keeping the older bulls, and I started looking when genomics first started to try and keep pace until we could get genomics up. I thought we have to go generation as fast as we can possibly go, try and get our new sires out of young cows, and that's basically the way we've gone. So we've sort of implemented a bit of genomics without the measurement, so it'll be just great to have those ratings, and especially now too we're concentrating more on health traits, and that's what genomics will give us. So that's where we're trying to take it.
So just to finish up where we are, just wondering, this is a difficult one for you, Lyndon, but for others that, and just starting, are there some new traits? Are there things that through genomics you're able to look at, or ones that you actually would like to see? And the service delivery you mentioned about now, weekly runs, things like that. Is there any of those things that would sort of the next movement in genomics and delivery that would further enhance what you're already doing? So, Paul, do you want to start if there's anything, or is it too early in the journey?
So probably a bit too early in the journey for us, but yeah, no, I mean, as a breed, the Aussie Reds are definitely very data focused and thirsty for data too. So yeah, we've always been on the front foot with supplying as much data as we can to Datagene, I suppose, to try and showcase, I guess, a lot of the stuff that you can't see that's inside a cow. Yeah, whether we're going to get enough reliability for genomics on that it's going to be the tough one.
Okay, Trevor.
I'm probably thinking on the beef side of it, because there's so many dairy bull calves, or the fact that what the weight gains could be on them, on dairy bull calves, Holstein bull calves. We see the Angus have grass-fed and lot-fed on their genomic test. Do we look at that part of our system? Do we use a beef sort of to a certain level of growth quicker? But I see the health traits like this are all around the world, but new ones coming along, so it's only a matter of time, I think, before Australia will have them. So.
Okay, Sandra?
Probably lactation persistence on 2 levels. Frustrating when you can get certain cows, bulls that get to 200 days, pack their bag and say, see you, we're going on holidays. They don't really fly very well. And now with different milk factories are wanting flatter production, it'll be good to see cows that go up and maintain their production to their nutrition and just continue producing until near the end of the lactation. So that's probably 2 or 1 with 2 streams that would be pretty interesting.
Okay. And Linda, I'll give you a chance. Maybe you can get ahead of the pack and provide a couple of things that might be important to you that they can start incorporating from day 1 instead of day 10.
Well, probably the survival of the breed around the world, it's kicked off the last few years, is niche marketing and the manufacturing of specialty goods out of Guernsey milk. It's the most unique milk of cows. And looking at that going forward and protecting that, it's high in beta-carotene, and maybe we need to do some work in testing in those areas to enhance, because it is a great manufacturing milk, to make sure we really enhance that product for the breed long term.
Great. So, Iona, is there anything you'd like to finish with?
No, that's great. Thank you very much to everybody.
So I just on behalf take the opportunity to thank Iona for joining us remotely and the work's been done there, and also thank our panellists that have provided a really great cross-section of where things are at from the farm level. And so if everyone would like to thank our panel, that would be great. So you're now